Tyrosine metabolism. In this video, I'm going to talk about tyrosine metabolism and how it is associated with the genetic disorder. So as you can see, this is the structure of the tyrosine, and it's an amino acid, and it's very important in our body because many biosynthetic pathway requires this tyrosine. So thyroid hormone biosynthesis require tyrosine. So the precursor for thyroid hormone is actually the amino acid tyrosine, and the catecholamine biosynthesis also require tyrosine. Catecholamines means the norepinephrine and epinephrine. The dopamine, which is an inhibitory neurotransmitter, also require tyrosine for production. However, melanin production also depends upon tyrosine. That is why we should study the tyrosine metabolism, especially the tyrosine catabolism and the enzyme involved in this catabolism process, and what happens if these enzymes are mutated, and what disease we should develop if these enzymes are mutated, the genetic disorders associated with these enzymes. So just back to the steps. So first, what we have here, it's phenylalanine. Phenylalanine, you can see the structure of the phenylalanine. It has a phenyl group, so it's a benzene ring phenyl group again. So phenylalanine would be converted to tyrosine by phenylalanine hydroxylase. It's a hydroxylase enzyme. It will put a hydroxyl group here. As you can see, these two structures differ only in the hydroxyl group. So this phenylalanine hydroxylase will hydroxylate phenylalanine to convert it into tyrosine. In the next step, tyrosine would be converted by tyrosine transaminase to para-hydroxypyruvate, and any problem in this step will cause tyrosinemia too. Just one thing to remember, that in this first step, that means the conversion of phenylalanine to tyrosine is very important. So phenylalanine to tyrosine is converted by phenylalanine hydroxylase, and any problem in this enzyme, if this enzyme is mutated or not working properly, we will develop phenylketonuria. And back to the thing. So we have para-hydroxypyruvate, para-hydroxyphenylpyruvate actually. So this phenylpyruvate is converted to homogeneous or homogenistic acid by beta-hydroxyphenylalanine dioxygenase. So it's a dioxygenase enzyme. It will decarboxylate this pyruvate, and it will form homogeneous. This is the structure of the homogeneous, and any problem in this step in this enzyme will ultimately lead to tyrosinemia type 3. Again, homogeneous get converted to mallyl acetate. Actually, these rings break, and it forms the mallyl acetate, which is a linear structure, and this reaction is catalyzed by very important enzyme, that is homogeneous 1,2 dioxygenase. Homogeneous 1,2 dioxygenase. This is a dioxygenase enzyme, and this enzyme is very important because if this enzyme is mutated or this enzyme is not functional or not present, then we will develop alkaptonuria. That means production of black urine. So from mallyl acetate, it will be isomerized to fumaryl acetate, and this step is catalyzed by an isomerase enzyme. And from fumaryl acetate, fumaryl acetoacetate actually breaks this fumaryl acetate to acetoacetate and fumarate. You can see acetoacetate and fumarate, and this step is catalyzed by fumaryl acetoacetate. Any problem in this enzyme will ultimately lead to tyrosinemia. That is why this whole catabolism pathway from phenylalanine to these fumarate and acetoacetate is very important, because almost all the enzymes involved in this step, if they are not functioning properly, then it will lead to some disorders. Actually, these disorders which are associated with these enzymes, has been those metabolic disorders, are actually genetic. Because the gene for the enzymes, if the gene for these enzymes are mutated, or mutations in these gene occur, then functional enzymes would not be produced, and this whole pathway will not be functional. So that is why those diseases are developed. Hope you liked it. Please subscribe and comment.